how to run educational programs. Right, off you go now. It's recording.
Okay, so let me start. Right. So as I was saying, in the past, as I said, with longer 454 sequences, people did explore using multiple variable regions. Problem is when you pass through one of these conserved regions, as I'll talk about, you increase the rate of formation of something called PCR chimeras. So in general, particularly with Illumina sequencing, to me at least, I think targeting just one variable region makes sense. And by the way, if anybody has any questions, if anything's confusing, please ask during the talk, but also you can ask at the end. All right, so what I've done here is just taken a load of 16S sequences, actually from the tutorial that you're going to look at today, just to give you a sense of what the actual sequence data looks like. Okay, and actually it's always a good idea to look at the sequences. When I suppose this is quite a sad indication of how much bioinformatics I was doing even 10 years ago, but 15 years ago I did my first 16S sequencing, and I just looked at the sequence and I could immediately tell it wasn't 16S because of the frequencies of the different base pairs. And as it turned out, the sequencing center had accidentally sent me someone else's sample of human DNA sequence. And that just sort of shows how if you spend your whole life looking at sequence, you can get an understanding of what it should look like, right? So anyway, so this is a load of 16S sequences. These are forward and reverse reads of 250 base pairs on the V4 region. So the conserved primer for the forward reads is starting over here, and then I've got a few of the reverse reads where the primer is starting over there. But these reads almost entirely overlap. If you do a longer region, your forward and reverse reads won't entirely overlap. They'll perhaps just overlap over in the middle portion, but you do want to get some overlap. And actually, so this is from a microbial community, actually an anaerobic digester. But you can see how there is variation in the, how different base positions have different degrees of variation in sequence. So here we've got actually quite a conserved region, but here it's far more variable, okay? So this is just something to bear in mind in 16S data, and it's something that the algorithms will take into account. All right, so what can we do with 16S? Well, perhaps not that much, all right? Essentially, you can use it to profile the community. And the obvious way to do that is to take your reads and classify them against a database. So for my benefit then, how much taxonomic classification have you done earlier on in the course? Have you talked about taxonomic classification?
No.
Okay. All right. Well, that's a good. But probably because most of you have a biological background, you do understand what taxonomy is. The thing to bear in mind with taxonomy is it is literally just a system for classification. In an ideal world, it would reflect evolutionary history, but actually, probably as Mark will talk about a great deal, there is no reason why it should reflect evolutionary history.
I would say
if you
completely
want,
the opposite, Chris. It must reflect evolutionary history, otherwise it's illegitimate.
Okay, all right. Well, I suppose I take a more pragmatic viewpoint. Okay, I accept that we live in an imperfect world where our taxonomy does not necessarily reflect evolutionary history. If you want something that definitely reflects evolutionary history, then you need a phylogeny. A taxonomy is a labeling scheme.
For labelling nodes in a taxonomy.
Pardon?
For labelling nodes in a taxonomy, in a phylogeny, I mean.
Okay.
Anyway,
Well,
all right, we're
all right.
getting off topic. You say what
Yeah,
you're
yeah.
going to say.
I think we are. The fact is, there is, well, so this is what I'm illustrating. This figure here, it's very old. It's taken from 2006 or something, but it's showing different taxonomies as a Venn diagram, and it's looking at the phyla labels in those different taxonomies and the extent to which they share phyla names. So if they do reflect evolutionary history, only one of them is getting it right because they all disagree with each other in terms of their labeling schemes. I would prefer to say that you can't necessarily assume that a taxonomy definitely reflects evolutionary history. If you want evolutionary history, look at a phylogeny. Anyway, that's something that perhaps we can debate later on. Okay, so you have this taxonomy. It's a hierarchical naming scheme. I'll talk a little bit more about it in a minute. If we want to assign a taxonomy to our sequences, we just need a database of reference sequences with those labels on them, and then we're going to use that as to enable us to classify our reads against that taxonomy. And the key thing is it's a hierarchical labeling. So in the classic system, you've got kingdom, phylum, class, order, family, genus, and species. And here's an example. This is actually an edible mushroom, a porcini. It belongs to the kingdom of fungi. The phylum, well, I'm not going to try and say it, Basidiomycota. And its genre and species name is Boletus edulis. So that's actually the porcini. But that's a taxonomic classification. Actually, a lot of the functional databases have a hierarchy as well, and I'll talk about that a little bit more in a minute. So in terms of 16S databases, probably the most representative one is Silver. That's got 9.4 million 16S sequences in it, which is sort of incredible to me because as I said, I've been doing 16S analysis for at least 15 years. And when I started, I think it had 80,000 sequences. So yeah, it's now getting rather comprehensive. I think there is an alternative database, which is Green Genes. But I believe that Silver is one of the best curated. So yeah, that's probably a good starting point for 16S databases. So how do we actually classify the sequences? Well, we have to search the sequences against the database, and we do that actually using the sequence itself, and that's known as a homology search. And this is something that you've talked about previously, isn't it? You did this last week, right? So have you done sequence alignment?
Yes.
Okay. All right. So the important thing to bear in mind when you're doing these searches of 16S sequences against a database is that you want something called a global alignment. All right? Because the entire 16X sequence, so if the thing you're looking for is in the database, then you should see its entire, the entirety of the 16S region that you have sequenced. Okay, so that's why you want to do a global alignment, because the whole of the query sequence should be there. In what situation then would you use a local alignment? So this is a question for the audience. So in one of the applications that you did last week, when we're using a local alignment?
Chris, I wasn't there last week. What is a local alignment?
All right, so a local alignment is when you're only matching a portion of the query against the reference, not the entirety of the query against the reference. All right, well, look, it would be appropriate for when, say, you were aligning two genomes from the same species against each other because only portions of them will align. It would be appropriate, say, when you were trying to do a homology search for a particular protein against another genome. A local alignment is appropriate when only part of your query should be matching against your database. All right. So there is actually a perfect algorithm for doing global alignment. That's the Needleman-Wunsch. Unfortunately, it's rather slow. So typically, in practice, what you'll be doing in terms of this bioinformatics is a fast method for getting an approximate match, and then you might be doing slow Needleman-Wunsch on all those approximate matches to get the ideal alignment of your query sequence against the database. And that's actually what vSearch, for example, does, and also uSearch, which are probably the two most commonly used programs for this particular problem in 16S. Once you've got your alignment, you turn that alignment into something called an edit distance. So that's telling you how many changes in terms of sequence are necessary to turn your query into your database sequence. So here we see we've got 2 substitutions and 1 gap in comparing our query to the database. So the edit distance, the number of changes required to turn the query into the database is free. And then you'll get other statistics, such as the E-value. That basically gives you an estimate of the log probability that you could expect a hit this good by chance when searching a database.
They know all about that, Chris. They know about E-values.
Okay. Well, I don't think it's...
They know. They know about blast searches.
Yeah, but it's fine. You can repeat.
Yeah.
Okay. Well, would you use a blast search for questioning querying a 16S query against a reference database then? So that's a question for the audience. Would a blast be appropriate for that purpose, since they know all about it?
Hands up if you think yes.
Pardon?
Do you think yes? Any hands up? Do we think no? Hands up no.
There is 2, 3, 4. No.
There's 5 people would say no. Everyone else is unsure. Chris?
Okay. Well, I suppose in a way you're all right. Okay. No, actually no, it's not appropriate. It's definitely wrong. Okay. And I'll tell you 2 reasons why it's wrong. Okay. Firstly, because a blast is a local alignment. And as we just talked about, for 16S searches against a database, you want a global alignment. Okay. You may be able to fiddle the parameters on a blast search to force it to be pretty close to a global alignment, but that's not ideal. Okay. The second reason you wouldn't do it is because it's slow. Okay. It will take you a very long time to blast all your sequences against a significant sized database. And as I've just said, vSearch or uSearch, they do global alignments, so they're entirely appropriate for 16S, and they do it much quicker than blast. Yeah. Okay. So that's the type of bioinformatics that we would be using for this purpose. Typically you get back the top N hits. Now, often, and I think this is true of the data pipeline that you're going to do later, you simply just assign your sequence to the top hit. And that's actually probably reasonably good for 16S. You can do something slightly more sophisticated where you would take the top N hits, or the top hits which are above a certain threshold. You set 95% identity and take all the hits that match that, and then perform something called a lowest common ancestor on those hits. So you basically move up your taxonomy until you have a consensus agreement on the assignment. So if you disagreed at the species level, you wouldn't assign at the species level, but if all the hits came from the same genus, you would assign at the genus level. The problem with this, though, is that most taxa in a microbial community won't be in the database. That was actually true when I first wrote these slides in 2014. Thinking about it with silver, possibly for the human gut, a lot of your sequences now the majority of your sequences may well be in the database, but certainly when you go to a novel community, that's not going to be the case. So what can we do then? So essentially, and this is also something else that you've probably come across in previous research, we use something called operational taxonomic units. Now, taxonomy is a classification. We've got a set of known, it's a classification problem. We have a set of known things and we're assigning our sequence to that. Operational taxonomic units are a clustering problem. We're trying to find the de novo clusters of sequences in our data set on the basis that they should correspond to relevant biological entities. And the key thing here is that we can then do that without a reference database. So it doesn't matter if we don't have, it doesn't matter if we haven't even sequenced this community before, all right? So the simplest strategy for forming OTUs, and this is what people used to do, say, 10 years ago, is something called a hierarchical clustering. So here we've got 10 different sequences, and I've actually put taxonomic labels on them. Most of these sequences would be labeled to Chlorobium genus, but one here is to a related genre, Chlorobaculum. So we align all our sequences together. You would do a multiple sequence alignment for that. Have you done multiple sequence alignment as well as pairwise alignment?
No.
Yeah, they did.
Okay.
Well, they touched on it.
All right. I mean, multiple sequence alignment is just basically where you have n sequences and you want to align them all together. The problem with it is, as I said, pairwise alignment is slow. Multiple sequence alignment becomes even slower, and actually it becomes even less reliable. So yeah, this is what people were doing 15 years ago in 16S analysis, was giant multiple sequence alignments, which is definitely a bad thing. Then they moved on to doing pairwise alignments, which makes a lot more sense. From those alignments, you calculate a distance matrix based on those edit distances that I was talking about. So now for every sequence, we know how different it is from each of the other sequences. And then we can do a hierarchical clustering. So we join the first two sequences together, form a node in our clustering, and then join that to the next closest entity in the tree. And that forms a tree, and then to form OTUs, we define a particular cutoff. In this instance, it's 3%. Typically, 3% is considered to be equivalent or close to species, although actually, if you think about the full length 16S gene, it probably comes down to about more like 1.5%. But on a variable region, 3% generally is a good approximation to species. But you won't expect that your OTUs exactly match onto species. And in a way, it doesn't matter. The fact is, you want your OTUs to be biologically relevant units, not necessarily species. It could be finer resolution, or it could be a lower level resolution. It doesn't really matter. So yeah, as I said, pairwise alignments reduce a lot of the errors that are brought in by doing these large-scale multiple sequence alignments. Now, that worked for 454 data, where we might have 100,000 sequences in the study. These days, when you're doing MySeq data, you've got more like 1 million, and it's generally taken that hierarchical clustering is too slow, although you probably still can do it. Instead, people have moved on to something called centroid clustering. So in a centroid clustering, the key thing is it's a one-pass clustering. So we only have to look at each sequence once. And it's pretty straightforward. You take your first sequence, and it forms a centroid, a cluster center. Then you take your second sequence. If it's within a certain distance of that centroid, it gets assigned to it. If it's outside that cutoff, which is shown as T in this diagram, it forms a new centroid. And then you just go through every sequence in your data in your data set. Now you can sort of see that will cause problems. It means that your clustering will be dependent on the ordering of your sequences. Reorder your sequences, you'll get a different clustering. But yeah, it gives pretty good results. And almost all the OTU clustering programs would discard singletons as likely to be noise. Now, when I started in 16S analysis, this is the sort of thing people were doing. This was based on the old GS20, so this paper is from 2007. And basically, wherever people went, they found vast amounts of microbial diversity. So this is a study done by Mitch Sogin in I think this was deep sea vents. These are the number of 16S sequences that they obtained, and on the y-axis here is the number of OTUs that they obtained. Now this is something called a rarefaction curve, which we'll talk a little bit more about on Friday. So rarefaction curves are a way of deciding whether you've done enough sequencing. So whenever you go to environment and you do more sequencing, you'll see more things. With rarefaction, you consider how many OTUs you would have seen at a smaller amount of sequencing. And then you'll get this curve. And if the curve is starting to plateau, you can say we've done enough sequencing to access the majority of the diversity in this community. So they were finding tens of thousands of both bacterial and archaeal species, or OTUs, at these deep sea events, and the curves hadn't even saturated. So they coined this term the rare biosphere. They got all these exciting nature and science papers. Now, I began working with this data in 2007, and actually on one of their papers, they'd sequenced something called a mock community. So this is where you have a known set of organisms, and you can use it for validating the algorithms. So in their mock community, they had 50 species. I actually went to the back of one of their papers, took these published mock community sequences that they had. I repeated their bioinformatics pipeline precisely, and I found 1,000 OTUs in that mock community. And it was one of those, you don't get many moments like this in science, but I was genuinely flabbergasted, okay? I remember actually sitting in my office right now, looking at the output of that analysis, because I really expected to see, say, 60 or 70, because I knew there was noise, but I didn't realize that the noise was going to inflate the diversity by a factor of 20, and also I didn't realize, I was surprised that they'd never done this themselves, okay? They'd generated the data a year and a half ago. It was their bioinformatics pipeline. It was like, why didn't you actually test your pipeline? Okay, but anyway, so that was quite a shocking discovery, and it really called into question everything that these people were publishing. So yeah, so I published one of the first papers. There was another one that came out, I guess, slightly later actually. So I was the first by a month or two. Pointing out that actually these studies that took no account of sequencing errors when they formed OTUs were greatly overestimating the amount of diversity in the community. So I published that along with an algorithm for removing the errors from 16S sequence. And that's the critical thing here. If you're going to do anything with diversity, then you need to correct for sequencing and PCR errors. Now, nowadays, my algorithm is not used at all, and that's because it was designed for 454 data, and it was quite specific to that data type. These days, probably the most used algorithm is something called DADA2, and that's what you're going to do in the tutorial later on this morning. So we'll talk a little bit more about error correction for 16S and DADA2 in particular. So what's going on here? So the principle is in our sample, we've got different sequences from different species, and that's shown in these 4 different colors here. And they occur in different frequencies, reflecting the different abundances in the community. Although, as we will talk about, once you've done PCR, the abundance of the post-PCR products does not necessarily faithfully reflect the original abundance in the community. So you do PCR and then you do sequencing. But there's always the chance of random errors. And if you think about it, you're then more likely to see more errors around more abundant sequences because you're basically getting more copies of those. So if errors occur randomly, you'll see more errors are associated with those sequences. So that's what we're showing here. All the red dots here are, that's the actual true sequence, but it's surrounded by a cloud of errors. And that's true of the blue and less so for the green because it was less abundant to begin with. And what happens is if one of these errors is outside of your cutoff for defining a new OTU, then it appears like a new species that isn't actually, or it's a new unit of taxonomy that isn't truly in your data set. So yeah. Oh, and just to give you a little bit more background about the nature of these errors. So they come from the sequencing machine. So MySeq is not perfect. It has, well, I think about 0.05% error rate these days. But also the PCR process is not perfect. So the actual process of amplifying up your sequence will generate errors of 2 forms, in fact. One will be single base errors, which will be similar to sequencing errors. But the other will be these things called PCR chimeras, which I mentioned earlier. So a PCR chimera is formed during the actual process of PCR. Now, if you probably all have a sort of reasonable understanding of how PCR works. You have multiple rounds of amplification. If you get incomplete amplification, then you'll have a partially amplified sequence that can then form a primer in the next round of amplification. But it might form a primer on a different sequence to the one it originally came from. Okay. The result is then that you get a bit of sequence which looks like one of the true sequences for the first portion, and then matches to a different true sequence for the second portion. So this is why they're called chimeras, after the mythical beasts of antiquity, which was, I think, a chimera was part eagle, part lion, anyway, it was a mixture of monsters, okay? Well, a mixture of animals formed this monster called the chimera. So that's the other sort of error that you'll be wanting to remove from 16S data, both these sequencing errors, but also PCR chimeras. Now, the other thing to bear in mind is PCR bias, okay? So this is taken from a study of mock communities that we did a little while ago, but the principle holds even though the technology has changed. So we were doing 454, and we've got these different variable regions here, so V3, V4. We've also actually got metagenomic sequencing in this study, and then this is the true community, which was actually completely even. Now, yeah, so every species should have been present in the same abundance, but that's not what you see. So for this region, V3, V4, we see far more than we would expect of these organisms. And this is caused by PCR bias. So there's no such thing as a universal primer. Whenever you've got a primer, it's going to match some species better than others, okay? Also, GC content is important, okay? So some sequences in your community will amplify up better than others, and some will be entirely missed. That's what we're showing these blue regions here. These are a load of species that were missed by all these primers, okay? So that's important to bear in mind. Your 16S sequencing gives you a distorted view of the community. Metagenomics is better, but it's still not perfect. And that's due to DNA extraction biases. Oh, and there's also another factor in 16S sequencing, and that is the fact that the 16S gene occurs in multiple copies. And so some species will have more copies of that gene than others, and so that will also distort their abundance in terms of your sequencing. Now that's something you can deal with if you've got a really good database of 16S gene copies, but as we talked about, that's hard to obtain. So yeah, so there's all these sources of bias, and in fact, there was this point in about 2010 where people discovered this, and I even went to a lecture by somebody. This was actually the chap that does USearch, and he concluded he'd quantified these biases just as we had, and he concluded his lecture with, so there is no point in doing 16S sequencing. Okay, he was literally like, you should just stop. All right, because you know it is such a biased reflection of your community. Okay, but actually that doesn't matter for a lot of the purposes that you actually want to use 16S sequencing for. And the reason is because although it's biased, it's still quantitative, and I'll explain exactly what I mean by that. So this is taken from our example from mock communities that we were looking at. Now, on the x-axis, we did two communities, one with even abundances and one with uneven. And on the x-axis, I'm showing the ratio of abundances between the true ratio of abundances for the species in the uneven versus the even community. And on the y-axis, I'm showing their predicted ratio from the 16S sequencing we did. And you can see that there's a very good correlation between the two things. So what this is telling you is that even though the 16S sequence is biased, those biases are consistent across samples. You can use it to understand how the relative to the rest of the community abundance of an organism is changing. And that's basically, why it's a useful procedure that can help you understand how community structure changes. Because as I said, it's very biased, it's full of noise, etc. But it is essentially quantitative. Those biases are predictable. Okay, so how does DADA to remove errors? Well, DADA stands for Divisive Amplicon Denoising Algorithm. So what it basically does is it starts with the most abundant sequence in your data set. It says that is real, no problem. Then it goes to the next most abundant, and it considers the closest match to that you've decided is real. And it has a simple Poisson error model, and it calculates the probability that you could have generated that sequence from one of the sequences you've designated as true through chance, giving an error model that also takes account of sequence quality, etc. And then it just moves through the sequences, deciding, is this noise, is this error? So yeah, this is actually why I didn't adapt amplicon noise to my seq data. My algorithm was iterative. DADA, it was much faster because it said it's sort of a bit like that partitioning algorithm. It only has to pass through the data set once. And this is showing, basically DADA working in a perfect fashion. So here on the y-axis, we've got the number of the observed reads at a certain edit distance to one true sequence. So we see a lot of sequences with just one error, but then less with two. That's the nature of a Poisson noise. And what DADA is trying to do is to define this line here, where we can correctly discriminate sequencing errors from true variation. So these red dots correspond to actual other true sequences in the sample. But it can't work with singletons. It basically ignores all singletons. And actually that will mean that for low depth sequencing, you'll probably underestimate the true diversity using DADA. Okay, So we can denoise our sequences with Dada. And a result of this is actually people have suggested that we could move away from OTUs entirely. So this is the concept of amplicon sequence variance. So the idea here is that you don't bother forming operational taxonomic units. You just take your sample sequences, you get your reads, and then you reform what is called amplicon sequence variance. And assuming Dada has worked perfectly, they should correspond to your original sample sequences. So the idea is, rather than forming OTUs for your analysis, you work directly with these ASVs. And there could be some advantages to that. If you think about it, you've then got the maximum level of taxonomic resolution. There are reasons, though, why I still prefer, well, I still think there could be some advantages to forming OTUs after forming your ASVs. And that's because OTUs give you a well-defined measure of sequence diversity. So if say you're interested in how many different things are there, if you're looking at ASVs, you're counting even a one base change as a new thing. Whereas if we form OTUs and count the number of OTUs, we've turned it into a universal unit. So actually what we would recommend, and I think you'll be doing in the tutorial, is that you do it both ways. You form amplicon sequence variants and you do analysis with those, but then you also might want to form OTUs, say at 3%, and do analysis with those as well. And sometimes we find OTUs perform better, and sometimes we find ASVs perform better. Okay, so I'm sort of coming to the end of what I wanted to say in terms of 16S algorithms. There are some things I haven't really covered in much detail. I talked briefly about mock communities and how you can use those to evaluate these algorithms. And now there's loads of different OTU algorithms. One, for instance, I was involved with is Swarm, which is it's a complete linkage clustering. So if anything is within a set, so it's sort of to chains the OTUs together, and that might be more appropriate in some situations. There's a lot of algorithms actually that attempt to be more ecological. So they also take into account correlation across multiple samples, which is a bit like the contig binning we're going to talk about later on today. So the idea is if actually you've got a sequence which is from the same ecological unit, then it might have the same profile across multiple samples, and then you can cluster those together and get something that's more ecologically informative. Now, 16S is limited. So the key thing is that the 16S gene is purely structural. So it tells you nothing about the function of a community, and that's why we want to do shotgun metagenomics, which we'll get on to in a minute. But there are a variety of algorithms that attempt to infer the functional potential of a community from a 16S profile. So the most popular one is something called PyCrust. And the way PyCrust works is pretty simple. It has a big genome of databases with associated 16S. You assign your 16S against that database. Then you've got a profile of genomes, and you use that to reconstruct the total metabolic functional potential of the community. I was never very excited about PyCrust, as I'll explain later on today, because to me, the aggregate metabolic potential of a community isn't terribly useful anyway. What you really want to know is who is doing what in that community. And for that, you can't avoid having to do metagenomics and assembly and binning, as we'll talk about. But, you know, PyCrust is immensely popular. And I think in some communities where you've got, you know, like the human gut, where you've got a lot of genomes available, it possibly does tell you some useful things sometimes. So, yeah, I'm not saying definitely don't do it, but yeah, it doesn't sort of make me super excited. Now, there's now completely integrated pipelines for 16S bioinformatics. The one you've probably heard about a lot is Chime. But I also mentioned Lotus 2 because that's developed by Sol at the QIB. And I mean, possibly has some advantages over Chime. They've got some very careful filtering of sequences that improves, gives you better results beyond just doing GARDA by itself. Okay. And now I've just got a couple of slides showing you the results of 116S analysis to try and give you a little bit more context of what you can do with this type of sequencing. So this is a study of something called Crohn's disease that I've been involved with for a long time, actually. So Crohn's disease is it's a form of IBD, so inflammatory bowel disease. It's a chronic inflammatory condition of the GI tract, and it's actually increasing in frequency in the Western or in the developed world, and now also in the developing as they adopt more westernized diets, and we don't really understand why. Now, there's been lots of cross-sectional studies of Crohn's disease. So here, people took, lots of samples from control individuals that were healthy, people with Crohn's. They do 16S sequencing, and you can find organisms that are associated with Crohn's and organisms that are associated with the healthy controls. And actually, if you do a lot of microbiome research, these things are sort of definitely nasty organisms, are associated often with dysbiosis. And these things, generally look like good organisms. Faecalibacterium prausnitzii is a classic example of a butyrate producer that's meant to be good for you, etc. But a cross-sectional study is limited in many ways, okay? It only tells you associations. A major problem with it is that everybody has a different microbiome that is particular to themselves, and we don't know what component of that is being driven by Crohn's versus their own individual variation. So what I've been working on for many years now, in collaboration with Kostas Gerasimadis at the University of Glasgow, is the study of Crohn's where we actually look at people as they get better. So there is a dietary treatment for Crohn's. It's something called exclusive enteral nutrition. It's produced by Nestle under the brand name Modulin. And if you give people, and it's typically used for pediatric treatment of Crohn's, it's mostly used for pediatric treatment because adults refuse to take it, okay? It's basically an incredibly bland milkshake that, you can force children to have, but if you give it to adults, they almost certainly cheat, okay? You have to have this EEM for 8 weeks, no other foods, and it induces clinical remission in the majority of cases, 80% or so. So what we wanted to look at is how the microbiome changes during the course of this treatment. And we had 21 controls, and then around 23 subjects, and 4 time points from those subjects. Time point A, right at the start of treatment, B and C in the middle of treatment, and D at the end. And we also had an additional time point E when they are back on their normal diet. And as I said, we've been studying this for years now. The original CICRA data set is nice because we have a whole range of measurements from these fecal samples. We have fecal calprotectin that gives you an overall measure of inflammation. We also have short-chain fatty acids. These are things like acetate, butyrate, propionate. We have 16S sequencing that I'm going to talk about. We also have shotgun metagenomics and LCMS metabolites. Okay, so we published 16S results in 2015, and I won't go into them in great detail, but the results were very useful, I would say. And to me, as I said, you can sort of criticize 16S, but for profiling a community, I still think it's an effective strategy. So when we looked at these individuals over time, we could see that there was a clear decrease in diversity associated with the diet. Firstly, just to say there was, these were people on the free diet, H are the healthy controls. So you can see that at the start of the treatment, the people with Crohn's disease had lower diversity, but the actual effect of the diet was to decrease their diversity further. So there's a sense in which the diet isn't making them look more like a healthy community. Instead, what we concluded from this study, and the NMDS plot here shows the same thing. So we'll talk all about the statistics of the microbiome on Friday, but an NMDS plot, and this is probably something you've definitely not covered up till now, right? So this is a means of ordination. So it's a way of taking a high-dimensional data point, in this case it's our 16S profile, and represented it in 2 dimensions. So the distance between samples, and each dot is a sample, in the NMDS reflects how different they are in terms of their actual microbiome profile. So yeah, if we look at the NMDS, we see the same thing. So this is group A at the start of treatment, and at B, E, and H is the healthy controls again, and A to B, they actually shift away from looking like the healthy samples, and E, when they're back on the free diet, they're closer to the, they're actually closer to looking healthy, ironically. So we're able to conclude from this that, the effect of the diet is not to shift you to a normal microbiota, it's doing something else. And actually the next, what we're currently working on is trying to understand what that something else is. And maybe I'll talk a little bit about that later. So look, how are we doing for time? Because I think that took a little while, didn't it?
Yeah, I think you should be closing the talk part now.
All right, well look, actually I've only halfway through it. So that was 16S sequencing. I also now have another 45 minutes on metagenomics.
Yeah, but you can do that this afternoon.
Well, that wasn't my plan, but I think that is a better way to do it, actually.
Good.
So we can now shift, I think, to the 16S tutorial, and then I'll come back this afternoon and give the second half of this talk. But before we stop, then, do we have any questions on any of that? There must be at least one question, or it's all perfectly clear, right?
No, they want to get their hands dirty. They just want to get on with it.
Fine.
We'll have some questions. There's one question.
No problem.
We got one question.
Chris, one question.
Hi, sorry. On the first diagram where you were explaining the operational taxonomic units, and the green sample disappears, that's because it's been classified in the same taxonomic unit as the red sample, and that's why that's gone.
Let's go back. Can I just go back to exactly what you're referring to? The green sample disappears. Is it this here, right?
Yeah, and that's because it's classified in the same taxonomic unit as the red one. So it's still in there.
Yes.
It's not been discarded. Okay, thank you.
Yeah, so that's actually sort of the motivation for these amplicon sequence variants. Actually, that's a nice question because I didn't explain this properly, did I? So when we form OTUs here, we have combined the red and the green together, all right? But when we go to this slide here, so when we, instead, if we use amplicon sequence variants, we would actually see these as 2 different things. So we've got higher resolution. So this is the sort of motivation for using ASVs, isn't it? But then, I guess what I was trying to say is that if you want to sort of count up diversity, it might be actually better to combine those 2 things. So I do sort of feel that there is, pros and cons to ASVs versus OTUs, whereas like Ben Callahan, Susan Holmes, they're very much of the view ASVs is what you should use. I would recommend both approaches. Not least because if you don't use ASVs, your reviewers will get upset. Okay, is that clear now?
Yeah, I think so.
So you group them together basically to make data processing easier down the line.
Not quite. Actually, you group them together because under the concept of OTUs, they are the same OTU. Okay. So if we're imagining that we're trying to form OTUs at the species level, those two different sequences would correspond to different strains of the same species. So if we want to profile species, we should group them together. Okay. Now, what the ASV sort of proposal is that we get the maximum resolution by not grouping anything once we've removed noise. So when people weren't removing noise, it was sort of like, well, we'll group them, we have to do some grouping to try and reduce the overall noise level. Okay. The data sort of viewpoint is our noise correction is now so good that we can use every error corrected read as a valid observation of something. Okay. But in terms of OTU formation, you are still joining them together because you think they are the same organism at the level of resolution that you're interested in. Okay, is that clear now?
Yeah, thank you.
I mean, OTUs, I've been to like week-long workshops on OTUs. You can debate OTUs.
You're fighting the last war here.
Well,
this is something that is in the dustbin of history by now, really, for anyone at the cutting edge.
Okay, well, I totally disagree with you.
I know. That's why I'm provoking you.
The point is,
stop
and this is
aggravating him,
something that I have, this is like something I've accumulated through bitter experience, right?
Yeah, but we don't do short read sequencing anymore.
Pardon?
We don't do short read sequencing anymore
Of 16S?
of anything.
No one, you're trying to tell me no one does 16S sequencing of anything that's just utter cobblers, Mark. Just go and count the 1,000.
Yeah, I know.
papers
I'm not
published
saying
last
that.
month.
No, there's a difference between are and should. Anyway,
No, actually,
this is
that is worth debating.
Yeah.
Okay. So, and I have this every time I talk to a group of collaborators about how to do their microbiome sequencing. They will say to me, "Do we need to even bother doing 16S because we're doing shotgun metagenomics?" And every time I say yes. Why not do 16S as well? Okay, because you will still get the community to more depth. Okay, because you're only targeting one read, one gene. So with a relatively small number of reads, you can actually get a greater depth of community profile than you'll get from metagenomics. Okay, plus, that was simple. Okay, it should have been simple. If those explanation of those algorithms was not simple, I did a bad job. Metagenomics bioinformatics is complicated. And there's a lot of sort of difficulties into translating metagenomics into a profile. So I still think that there is a benefit to doing 16S as well. A good study, in my opinion, combines 16S and metagenomics. So Mark, I completely disagree with you.
okay.
Okay. It's a shame I'm not there in person because we could debate that endlessly, no doubt.
Well, we can still debate it. The difference is you can't punch me now,
That's definitely a
which
good
is probably a
thing.
good thing. Yeah. Anyway, all right, I'm being provocative here, and I'm sure they understand that. Okay,
Yeah,
well,
no, but actually, Mark, that is something that's raised by almost every collaborator I ever have. It's
Yeah,
like,
yeah, exactly.
do
I'm not
we
being
need to actually do
unreasonable.
16S?
yeah.
I'm like, yeah, just why not do 16S? You can analyze 16S reads in an afternoon. It will take us months and months to analyze your metagenomes.
Yeah, but the
it would take
16S
from
won't
the
tell
view-
you anything about function.
Yeah, obviously, it's limited, and we'll talk about that this afternoon, but it's still a useful technique. It's like, to be honest with you, right, I used to once upon a time work on GGE, okay? And, that's probably still useful. It's better than nothing. It's not as good as 16S, but it still tells you something. And actually, I think there's reasons why, for some purposes, 16S is still better than shotgun metagenomics. Because it's got a greater depth and because it's less ambiguity in terms of what you're seeing. So yeah. All right. So look, we can unless there's any other questions, we can leave it there. That was a good
Okay.
question.
Okay,
Alright.
I'll
Okay,
stop
so look,
recording now then.